Welcome to Spring LIB.
Okay, so problem 60 is about the ways in which bacterium might sort of transfer their genes. Okay, and so the key to this one is just knowing what all these words mean, right? So if we want to look at transduction, so transduction is this process by which a bacterial cell here, and it has its DNA, right? There would be a bacteriophage, okay, that sort of injects it with a new little piece of DNA, right? And that causes let's make this one smaller. Then we get to see that the DNA gets incorporated into it, right? So that's sort of what's happening with transduction, is the bacteriophage transfers the DNA, and the bacteria then takes it up into its own DNA. And similarly, a transformation then, so if we look at transformation, okay, and in this instance, it's really very similar. So we have a cell with its nucleoid DNA, and instead of a bacteriophage injecting it, it's going to sort of take it in from the environment. But then the end result is going to be the same, okay, where that DNA piece is now incorporated into the host genome. Okay, and then if we go to next on the list is conjugation. Okay, so for conjugation, this one's a little bit different. This one requires that we have two cells. Okay, cell 1 and cell 2, and each with their own genomes. Okay, but one of them will sort of transfer their DNA via a mating bridge. So this would be called a mating bridge. Okay, but all these do sort of transfer DNA. So that sort of leads us to this last piece of binary fission. And binary fission is actually the process by which prokaryotic cells duplicate. Okay, so it's just the sort of process of having one cell and the one DNA, and then that DNA will sort of start to duplicate, right? So it starts to duplicate in both directions. Okay, and eventually you sort of get an elongated cell with these two pieces of DNA, and then that will pinch off into two daughter cells that are identical, right? And so the idea here is that if this initial cell is resistant, okay, well, probably both its daughter cells are resistant, but these daughter cells are non-pathogenic because it is identical to the first one. So if this one is not a pathogen, so this one's non-pathogenic, then so are these other two, okay? And so the false statement then is going to be choice D.